Hello everyone, welcome back to Krishna's lectures. We shall now study cell division under the following headings. Introduction. We have to understand that the cell division is a part of cell cycle. Cell undergoes cell division and thereby completes its cell cycle. This is the most important statement, I would say. Okay, because when we talk about cell cycle, we have interphase and we have M phase. So this is the M phase and the rest is actually the interphase, because interphase lasts for more than 95% of the duration. So this M phase is actually called mitosis phase. During this phase the cell divides. So cell division is a part of cell cycle, correct? Cell undergoes cell division and completes its cell cycle. This is the most important thing and we have to understand this, that cell division is a part of cell cycle. And then we have two types of cell division. First is mitosis and the second is meiosis. We have to understand that mitosis is called as equational cell division and meiosis is called as reductional cell division. The M phase. This is the most important and this is what we are going to study today. M phase has been divided into two phases, karyokinesis and cytokinesis. Karyokinesis is divided into prophase, metaphase, anaphase and telophase. Cytokinesis is actually defined as the division of cytoplasm. Okay, so here what happens, we already have read about all these phases in the lower classes. So what we have to focus on is what is the beginning of each phase and what is the characteristic feature that is seen in this phase, and then what happens. So under these three headings we shall be studying all these four phases. See, there is no such division. There is no division when it comes to cell division stages. Okay, whatever the stages of cell division we have, per se there is no division. Cell division is progressive. There is no stopping in between. I mean, in meiosis we have, in female gamete formation there is stoppage. Except for that we don't have any stoppage in cell division. There is no proper boundary defining prophase and metaphase or anaphase and telophase. But for our understanding, it has been divided into stages and we should know when a stage begins, what are the characteristic features and what happens. Now prophase, metaphase, anaphase and telophase, all of them we are going to study together. Fine. We have a cell. So what is happening here, we are talking about karyokinesis. That is, karyokinesis means division of the nucleus of the cell. Cytokinesis is division of the cytoplasm of the cell. That both of these are together called as M phase. They both make up M phase, and at the end of this M phase there are going to be two daughter cells, both with 2N. Okay, two daughter cells are formed. Here first what happens is the nuclear membrane starts disintegrating, the nucleolus starts disappearing, and then all the organelles disappear like endoplasmic reticulum, Golgi apparatus, everything. So here I am just drawing a schematic representation of the nucleus dissolving. We have nucleolus, nuclear membrane, endoplasmic reticulum and Golgi apparatus. All of them disappear. This is first, and then we have the centrioles which are formed. These centrioles start moving apart. The centrioles move apart, and then we have the chromosomes condensing here. Chromosomes condense, and chromatin material, sorry, chromatin material, chromatin material condenses to form chromosomes. Here we have these mitotic apparatus being formed. Mitotic apparatus, when we say, it is these two asters and mitotic fiber. Both of these are together called as mitotic apparatus. We have chromosomes here. I am just drawing one chromosome. So whatever I am drawing is the equatorial plane. Here the chromosomes actually lie, and then exactly at the centromere position the chromosome is going to split. Chromosome is going to split into two parts, and then they start moving. This is the centromere and this is the centromere. They start moving towards the opposite poles. After this, all the organelles that had disappeared are going to reappear. So we would be having two nuclei, and then there will be a new furrow which is going to be formed, which will deepen, and then it will give rise to two daughter cells, both with 2n chromosomes. So we have nuclear membrane reappearing, all of those which had disappeared, nuclear membrane, and then we have nucleolus, and we have Golgi apparatus, Golgi bodies, and then we have endoplasmic reticulum, ER. All of them reappear back. So we have a chromatin material. Chromosomes lose their individuality. This is actually what is called as telophase. Chromosomes. When we talk about chromosomes, we have to understand that it has arms. This is a centromere. This black one is actually the one which is drawn in black is centromere, and centromere, and this red one is kinetochore. So the kinetochore is actually a protein which is in the form of a disc and is present on the centromere. This is going to be the attachment site for the spindle fibers. The movement of the chromosome is facilitated by these. We have a chromosome. I am drawing a very big chromosome here. This chromosome is attached to the mitotic fiber here, which is represented in blue. This mitotic fiber is attached to the centromere via kinetochore. Kinetochore is disc-shaped proteinaceous material. So this is the one which is responsible for the mobility of the chromosomes during cell division. Movement is happening because of this, that is kinetochore being attached to the mitotic fibers. We shall now study prophase under the three headings. There is no such division of stages during cell division. Cell division is progressive, but for the ease of reading it, we have divided, and we need to know when which stage starts and when it ends, and what are the characteristic features and what happens. Those three. Prophase, when does it begin? Initiation of the condensation of chromosomal material. The moment condensation of the chromosome material happens is when prophase is said to begin. What happens during prophase? In prophase, the centrioles move apart, mitotic apparatus is formed, and cell organelles disappear. How many? 4. This rule of 4 is going to help you understand the whole of cell division. I once again repeat, rule of 4 is very important. It is going to help you understand mitosis. There is no such thing as rule of 4. It is something that I have made it up, and that is for helping you guys understand it and easily remember. There are 4 things that disappear in prophase and there are 4 things that reappear in telophase. And what are the 4 things that disappear in prophase? We have Golgi, endoplasmic reticulum, nuclear membrane and nucleolus. All these 4 disappear. And what is the key event happening in prophase? Disappearance of 4, that is nuclear membrane, nucleolus, endoplasmic reticulum and Golgi apparatus.